Hi there, everyone. This is Drew Heinmiller from VisualSonics. I'm a product manager here at FUJIFILM VisualSonics, and we're joined today by Eno Hysi. He's a PhD candidate at the Department of Physics at Ryerson here in Toronto. So he's not actually with us in the room just now, but he is in the same city. So there's that. He's going to talk to us today about photoacoustic RF techniques for imaging cancer treatment response. Just before we get started, I've got a couple of housekeeping things to share. The first is we will be recording the webinar, and we'll be providing a recording of the webinar after the broadcast. All lines will be muted for the duration of the webinar, so if you have any questions, please use the Q&A button and just post your question at any time during the presentation. We're going to answer those questions at the end of the session. The talk today is going to be around 45 minutes or so, and then we'll have a little bit of time at the for questions at the end. So I'd like to introduce Eno Hysi. He's a PhD candidate, as I mentioned, at the Department of Physics at Ryerson here in Toronto. He's a Vanier Scholar. He received his Bachelor of Science in 2010 from Ryerson in medical physics, where he was introduced to photoacoustic imaging. He continued his research there, where in 2012 he graduated from his MSc in biomedical physics with a gold medal. And his research interests include photoacoustic tissue characterization for biomedical applications for cancer treatment monitoring, for example, blood hemodynamics, and kidney transplantation. And now I'm happy to hand it over to Eno. You can take it from here.
Excellent. Thank you so much, Drew, and I welcome everyone to this webinar here today. It's great to have the opportunity to share some of our work on imaging cancer treatment response using photoacoustic imaging, and specifically focusing on the analysis of the radiofrequency techniques that I will be covering. So before I begin, I want to give you a very broad overview. Hopefully by the end of today's talk, I have convinced you, or at least tried to convince you, of the fact that thunder, orchestras, and cancer tumors and their blood vessels are actually related. And allow me to start off with the first one. And if you're in Toronto or anywhere else for that matter, yesterday you would have heard a little bit of lightning followed by the thunders. So typically, the reason why I'm starting with this concept, because this is one of the main analogies of photoacoustic imaging. So just like during a thunderstorm, you first see the lightning, and shortly after you hear the accompanying thunder. Photoacoustic imaging is a hybrid modality that very much relies on the same principle. Of course, we're not illuminating, or we're not shooting thunder at anyone's biological tissue, but instead we are actually using optical fibers to illuminate a cross-section of tissue which typically contains an optical absorber. So this rapid illumination with a short laser pulse creates a very localized thermoelastic expansion of tissue, which generates acoustic waves, which then propagate and are detected by an ultrasound transducer, which typically operates in passive mode. And by acquiring these signals and reconstructing them in various ways, you actually end up generating a photoacoustic image of this very same region of interest. Now, photoacoustic imaging is extremely successful, but because it relies on laser illumination at multiple biological length scales, you have the ability to actually probe as small of structures as organelles inside a cell, all the way up to an entire organ, as shown here in panel D, which actually represents blood vessels from a breast. In addition to being able to probe multiple biological length scales with very high contrast and resolution, photoacoustic imaging has the ability to provide functional information as well. And what I'm showing here in the bottom, and I'll talk a little bit more detail of how these images are generated, is an oxygenation map of a living mouse ear. And you can examine the exquisite detail that arises from the fact that you're dealing with optical contrast as well as with ultrasonic resolution within these images. And that concludes the thunder part of my talk, and hopefully I've convinced you that the analogy that I use of thunder and lightning is used to explain the photoacoustic effect. The next thing I will be talking about is the orchestra aspect of it. But before I dive into the orchestra, I want to talk a little bit about how photoacoustic images are actually formed. And what I'm showing here in this slide are some of the photoacoustic imaging transducers used in the VisualSonics Vivo laser system, which we currently have available in our laboratory. And each of these transducers more or less relies on the same principle. And that principle I'll explain to you through this panel here, going from A all the way to E. So follow along with me as I'm pointing towards each of these sections. So if we imagine a collection of absorbers. In this case, I've made a collection of spheres of varying sizes scattered randomly throughout space. The actual photoacoustic response from each of these three individual spheres, which I've highlighted here, is your typical N-shaped signal. However, this N-shaped signal, as is detected from any of those transducers, undergoes something called band limiting, which is essentially another way of referring to the filtering of that very same signal. And the filtering of those signals, taking into account the frequency band of your transducer, will result in distortion or some distortion of the N shape, which when we account for all of the absorbers within our region of interest and perform spatiotemporal superposition, or also known as beamforming, will result in a signal that looks very much like this. And of course, because we have typically multiple linear array elements in our transducers, if we perform delay and sum beamforming and account for the directivity of these transducers, you end up with an image that is shown here in panel E. Now, what I want to emphasize, as alluded by the title of my talk, is the radio frequency signals. And the radio frequency, or RF signals, as I'll be referring to them here on after, is key to the type of analysis that we are performing. All of the photoacoustic images that I showed up until this point, they typically rely on the amplitude of the signal, denoted here in red, and they simply map this amplitude as a function of time or space in order to acquire depth resolved images. However, there are more components to this RF signal which are typically ignored when conducting photoacoustic imaging. And the ones that I'll be talking about today is the probability distribution of these signal amplitudes, also known as envelope statistics. And the other component is the frequency content of these signals. And those fall under the category of RF spectroscopy and Cepstral. And I will be explaining in the next slide the details of how these techniques are actually utilized. But what I wanted to highlight is the fact that these techniques are all very successfully used in ultrasound tissue characterization. And what we have done over the years is adopted them to photoacoustic imaging in order to characterize various aspects of biological tissues, as I'll show you later on with cancer treatment monitoring. So these RF analysis methods broadly are divided into two categories: the ones that rely on the temporal or time domain, and the ones that rely on the frequency domain of our photoacoustic signals, shown here. So the temporal domain approaches revolves around the envelope statistics, and the idea here is that you actually map the envelope uh, of the signal amplitudes of your photoacoustic signals, and you fit that to very specialized histogram distributions whose fit parameters actually encode information regarding your optical absorbers in this case. The frequency domain approach relies on now transforming your time domain signal in the frequency space, typically performing what is known as normalization, where you remove the dependencies of your transducer or the transducer response, which is typically shown by this blue curve here. And in normalizing that curve, you end up with what is known as the normalized power spectrum within the typically the 6 or 10 dB of your ultrasound bandwidth. And once again, relying on the fit parameters of this curve, typically the spectral slope being the most commonly used of them, you can actually obtain useful information about the structure and the concentration of your underlying tissue absorbers without having the need to resolve them. And the last technique is something that we have recently been working on, which is known as Cepstral analysis. And mathematically speaking, that simply is the inverse Fourier transform of the logarithm of the Fourier transform of your photoacoustic signal. And I know it's a mouthful, but mathematically speaking, it's actually written right over here. And the idea here is now you end up with something called the power cepstrum, which instead of plotted as a function of frequency, you're now plotting it as a function of distance. And the distance that I'm referring to is not necessarily the distance relative to the transducer as you would plot in a time domain signal, but rather the spacing between adjacent absorbers, assuming that there is some periodicity in their organization. And I'll show you more details as to how this can be used to probe those periodicities within your absorbers. So I'll start off with some simulation results with each of these where we highlight each of these approaches, so to give you a feel for the type of information that you can actually obtain. So I'll start off with the envelope statistics approach. And the envelope statistics approach relies on, once again, mapping the amplitude distribution of your photoacoustic signals for various concentrations, as I'm showing here in panel A. As we're going from left to right, we're actually increasing the concentration of these absorbers. And in panel B, what I'm showing is the envelope histograms that have been fitted to various models, from the simple Rayleigh to the generalized gamma and the Nakagami. And these are well-established models which actually provide information about the way that the distribution of your envelope of your signal amplitudes actually occurs. But what's most important, and this is where these techniques actually do become useful, isn't as much as the fitting of these parameters, but rather the fit parameters themselves. And in panels C, D, and E, I'm showing various of these fit parameters which have been plotted as a function of absorber radius, so another metric for size of your absorber, the number density of your absorbers, another metric for concentration, and a combination thereof. And if we say focus on the Nakagami omega parameter shown here in panel E, we see that this parameter is sensitive to both the size as well as the density of these absorbers. So right off the bat, I hope that I've convinced you that there are quantitative information contained within your signal amplitude, which can immediately provide you some information regarding how many absorbers you have within your resolution volume and what is their size. And some of those parameters are independent of size, as shown here by the Nakagami N parameter, and some parameters such as the generalized gamma A parameter are dependent on both size as well as your concentration. So that wraps up the envelope statistics aspect, and now I will dive into the radio frequency spectroscopy, where now we are actually using the frequency information of our photoacoustic signals to retrieve information about the sizes of our absorbers. So in panels A and B, what I'm showing here is representative. These are simulated images of collections of absorbers with various sizes, the size denoted on top of each image. And on panel B, I am actually keeping the average size of these absorbers the same and changing their polydispersity, or in other words, you can think of it as the variance within the sizes of our absorbers. The quantitative parameter that arises from this technique is called the spectral slope, or SS. And when I plot the spectral slope in panel C as a function of the bead radius, you will see that this parameter is capable of differentiating various sizes of beads up until typically the region of your transducer where the product of your wave number and radius of absorber approaches 1. And what's important to note is that this parameter is independent of concentration, as shown here for 2 different concentrations. So it is exclusively a size-dependent parameter. In addition to just monitoring the overall absorber size, we also have the ability to monitor the polydispersity, or the distribution in the size of the absorbers, as shown in panel D here, where by changing this theta parameter in our simulations, we are actually capable of monitoring the changes that occur as a function of increased polydispersity. So as the polydispersity increases, just like the absorber size increases, the spectral slope parameter is a quantitative parameter which decreases with increasing size. And once again, this relationship holds true for as long as your transducer frequency approaches a Ka equals to 1. And now I will be touching upon the Cepstral analysis approach, which, as I mentioned earlier, is an approach that has the ability to differentiate periodicities and quantify these periodicities within your resolution volume. So to simulate that, what we've done is we've simulated collections of absorbers, which were first placed in a lattice-like fashion, shown here in the leftmost figure of panel A. And just so that you follow along as I go through the rest of the slide, what I do here is I quantify what percentage of my spherical absorbers are in a grid fashion versus what percentage of them are in a random fashion. As we go from left to right, what I'm doing is I'm increasing the percentage of optical absorbers, or spherical absorbers in this case, which are placed in a grid or a periodic fashion, and increasing the percentage of absorbers which are randomly interdispersed through this medium. And by the time that you get to the third panel, most of our absorbers are randomly distributed. And the representative photoacoustic images acquired with a linear array mimicking the LZ250 Vivo laser probe are shown below. And you can see that as you switch from the lattice-like configuration all the way to the more random configuration on the right-hand side, the photoacoustic images reflect that. Now, if we were to simply rely on the amplitude of these signals and just observe the changes that are occurring from one image to the other, we wouldn't be able to obtain a lot of information until we switch to the Cepstral domain. So the Cepstral approach is capable of now differentiating between the adjacent spacing of our periodic absorbers, and the location of this peak here, denoted by the blue star in graph C, tells you what is the average spacing between absorbers which are periodically situated. So I remind you again that you cannot actually see those periodicities in this graph. However, this technique is capable of differentiating them. And of course, we have validated this with multiple different levels of absorber spacings, or you can think of this as multiple different concentrations. And you can see the agreement between the estimated Cepstral spacing and the true spacing is relatively high, which tells us that this technique is capable of monitoring the absorber spacings in a medium where you are unable to resolve them with a large degree of accuracy. So with that, I sort of want to summarize a little bit the three techniques that I was talking about here by once again bringing this conventional photoacoustic signal, one which contains contributions from absorbers of multiple sizes. And now I will reveal to you where my orchestra analogy comes from. If you were to listen to an overall orchestra sound, say play Vivaldi's Primavera, you might listen and hear something like this. However, if I isolate the violin, the sound that you hear is different. And our analysis techniques, which rely on the RF signals that are acquired from our transducers, work very much in a similar way. By applying each of those techniques, you're now capable of isolating the individual sources of your signal, even though you cannot differentiate them by looking at the image itself. And that's really the gist of our approach here. And now I will illustrate a practical use of these approaches, which is that of cancer treatment monitoring. And I will start off by giving a broad overview of the clinical motivation behind our work before I dive into how these techniques can actually provide useful information. So when a cancer diagnosis is typically made, from the moment the treatment begins all the way till the outcome of the treatment, whether it's positive or negative, typically physicians prescribe anywhere between 12 to 32 weeks of continuous radiation or chemotherapy or a combination thereof. And the challenge with this approach is the following. In order to monitor the success or failure of these treatments, which is just as important, clinicians typically rely on your conventional CT, PET, or MRI images, which are spectacular in differentiating volumetric changes within our tumor. But the question that I ask is whether or not it would be possible to tell whether or not a treatment is working long before that tumor actually undergoes shrinkage. And the reason why is because the shrinkage of the tumor might not occur for quite some time. And by that time, the patient has most likely undergone through several rounds of chemo or radiation. And you can imagine the frustration and, of course, the costs associated with undergoing a particular cancer treatment and not necessarily knowing whether or not that treatment is going to work. We believe that photoacoustic imaging offers several advantages compared to the conventional and very expensive approaches. And starting with the physical advantage, which is that of higher spatial resolution. You are now capable, depending on what type of photoacoustic imaging you're performing, actually go down to submicron resolution. And that differs significantly compared to the much lower resolution of CT or MRI imaging, which is typically in the order of a few millimeters. Photoacoustic imaging has a lot of contrast and high contrast, which arises from the optical absorption of these chromophores, mainly the oxy and deoxyhemoglobin, which I will be touching in a bit more detail in the next slide. And in addition to that, because photoacoustic imaging uses the same ultrasound technology and transducers as your conventional ultrasound machines do, you have the ability to acquire co-registered anatomical information of the same region of interest that you're eliminating via ultrasound. And in addition to that, in addition to the high spatial resolution and contrast, you have the ability to now provide functional information with regards to the structure that you're illuminating. And this information can be as detailed as your oxygenation or a metabolic rate of oxygen consumption, which several groups have worked on, in addition to obtaining information about non-resolvable vessel sizes and morphology, which is most what I will be mostly focusing on for the remainder of this talk. Now, I talked about the contrast, and the molecular specificity of photoacoustic imaging relies on the fact that, fortunately, many of the dominant tissue chromophores, such as oxy and deoxyhemoglobin shown here, have very unique spectral features. And what we're showing here is how the optical absorption coefficient as a function of optical wavelength, and taking advantage of these optical features of our dominant chromophores. And of course, there's a lot more of them depending on the type of application, but the application that I'll be focusing on will probe the oxygenation of the tumor, so we rely on the oxy and deoxy absorption spectra to obtain specificity and functional information within our images. So another motivation behind using photoacoustic imaging for treatment monitoring has to do with the fact that vascular architecture plays a significant role in cancer therapy. This has been known for a number of years that tumors have leaky vessels and tumors are hypoxic. And hypoxia, of course, has significant implications with radiation therapy. And the leakiness of the vessels within tumors has also just as significant implications in drug delivery, because if you imagine that you're delivering a particular cancer drug intravenously, but half of your tumor is not receiving that drug because it almost leaked out right as it entered the tumor, you can imagine how that impacts treatment response. And of course, another quite as important parameter is that of metastasis, which as a result of the buildup interstitial fluid within the tumors, it actually causes cancer cells to now go into the systemic vasculature and metastasize elsewhere, and you end up with a secondary tumor, which often becomes quite as problematic as the primary one, if not more. So all in all, photoacoustic imaging has a lot to offer to cancer treatment monitoring, once again, due to its high contrast ability to obtain functional information, and most importantly, the fact that tumor vessels play a significant role in treatment monitoring. So I will be talking to you about two studies, one of which is the thermosensitive liposome therapy, and this was a multi-institutional collaboration between us here at Ryerson University and mainly researchers at the Ontario Institute for Cancer Research. And this work has been published and presented in several meetings. And what this work relies on is on hyperthermia-induced drug targeting. Now, hyperthermia-induced drug targeting focuses on addressing a very important problem in chemotherapeutic delivery. When chemo is typically administered to patients, chemo goes both into the tumor, which is where it's supposed to damage cancer cells, but in addition to that, it also reaches the normal and healthy tissues. So our collaborators at the Ontario Institute for Cancer Research came up with a solution to this problem, which is a thermosensitive liposome. And this thermosensitive liposome was named HATDOX, which stands for heat-activated cytotoxic. And the idea here is that you have a 200 nanometer empty lipid bilayer, which you encapsulate your release drug, in this case doxorubicin, into. And of course, perhaps the name is self-explanatory, but the idea is this. At 37 degrees or normal physiological temperature, the injection of this drug within the tumor vasculature as well as the surrounding healthy tissue will not prevent the drug from being released, thereby reducing the systemic cytotoxicity of the drug. However, as you increase the temperature of this drug, the temperature of the tumor and the surrounding tissues to about 39 to 42 degrees Celsius, this thermosensitive liposome has the ability to actually burst and release this payload, this therapeutic payload, in a very localized fashion. And you can imagine that if you're only heating the tumor, then all of your drug will end up in the tumor, whereas systemically the rest of the patient's body is not receiving any drugs. Now, what's interesting about this particular drug is because of how much of it is actually released per unit time as well as per unit space, as early as 2 hours post-treatment, what has been observed is a significant amount of bleeding. And this is what this window chamber model is showing here. A lot of bleeding occurring as early as 2 hours post-treatment. Now, the question that I asked myself when I first saw these images is whether or not we can use photoacoustics to monitor this treatment. So with that in mind, we designed an in vivo study where we inoculated EMT6 breast cancer cells in the foot pad of mice, which will then submerged in a water bath kept at 43 degrees in order to allow us to heat the entire tumor uniformly. Using the Vivo laser system, we actually performed photoacoustic, ultrasound, and power Doppler imaging in 3 dimensions, and we acquired these images before treatment, which was done 30 minutes before the injection of the drug. We treated these animals with the HATDOX formulation for 1 hour at 43 degrees Celsius in the water bath, and then we followed these tumors after treatment at multiple time points, and you'll see most of these time points are actually were chosen in order to focus and monitor the early changes within our vasculature, and we followed these tumors as late as 7 days. So to illustrate to you how we actually estimate the oxygenation of these tumors, we rely on the amplitude of our RF signals. And most importantly, we also rely on the anatomical co-registered information that the ultrasound images provide, which allows us to segment a region of interest from the ultrasound, which we then apply to the photoacoustic images. And using a fairly simple linear spectral unmixing approach, what we do is we generate these oxygen saturation maps, SO2 maps, which our group has developed an approach to, instead of just relying on the average value of these SO2 maps, we were interested in looking at the distribution of these oxygen saturation values, both as a function of tumor location in three-dimensional space, as well as a function of time post-treatment. And what I'll show here is some representative oxygenation maps for our treated Haddox tumors and our heated saline. So as we start off with a fairly well oxygenated tumor, at pre-treatment, as soon as 30 minutes after treatment, once we have administered our thermosensitive liposome, there's a significant drop within the oxygenation of these tumors, which is not observed within our control group, which is once again just an injection of saline. If we now quantify this with our oxygenation histograms, what we actually notice is that that drop within the oxygenation of our treated group remains or persists for the first 24 hours, and by 7 days, the oxygenation of these tumors, most likely as a result of the neovascularization of these tumors, increases for both groups, whether it's treated or untreated. But it's important to note that the untreated saline group does not undergo any significant changes in the oxygenation of these tumors in the earlier time points. Now, what about the radiofrequency analysis? So I mentioned to you that we can obtain information about the vessels within our tumors. Well, how do we go about doing that in an actual experiment, unlike the simulations that I showed earlier? So once again, our starting point is our anatomical ultrasound image, which then is used to segment our region of interest. And in order for us to remove the frequency response or the system response, what we do is we make an independent reference phantom and apply the same region of interest in order to match the acoustic and optical attenuation of our tumor. And if you now normalize the photoacoustic image against this reference phantom at all of the spatial locations, you can now compute the frequency spectrum, which looks something like this. The blue thick line shows the average spectrum, and each of these individual lines show the different locations within the tumor. And what's important to note is that if you'll recall from my simulation results earlier, from this spectrum, we can obtain a parameter called the spectral slope, which I showed earlier was sensitive to the size of these absorbers. And if we now normalize or take these power spectra and look at them as a function of time post-treatment and simultaneously examine the changes in our oxygenation as a function of time post-treatment, you end up with curves that look like this. So this graph, this scatter plot here looks at the relationship between the mode of our SO2. So this is a metric that arises from our histogram distributions of oxygenation, in addition as a function of the spectral slope being a parameter which is sensitive to the size of these vessels. Now, I can spend a lot of time here, but I'm not going to, because there's quite a lot going on. But the message that I want to leave you with here is that this cluster approach here allows us to differentiate between those animals that responded to treatment versus our untreated control, as well as, just as importantly, focusing on those animals that did not respond to treatment. So this approach allows us to visualize the changes or the drop in the oxygenation of the tumors at the early time points, in addition to the changes in the spectral slope. Now, the question you might be asking is, so what? And that would be the right question to ask. So what does it mean for this early drop in oxygenation to occur? Well, the success or failure of any cancer treatment, whether it's in a preclinical regime or later on in the clinical domain, is whether or not the tumor has shrank. Typically, caliper measurements or volumetric measurements of tumors are performed in order to see if the tumor shrank in size or if it decreased. And what I'm going to be showing here is how the change in the size of these tumors for our responders and non-responders behaves as a function of our imaging biomarker, which is the change in the SO2 plotted on the x-axis. So the message here in this plot is the following. Those animals that had a significant drop in the tumor oxygenation as early as 2 hours post-treatments were also those same animals that 1 month after treatment at our endpoint shrank in tumor size. The non-responders were those animals whose size increased and oxygenation did not decrease. It relatively stayed pretty much the same as 0. And if we look at our heated saline control, once again, our non-responder behavior mimics that of untreated animals, whose size once again increased by an average of about 50%, and whose oxygenation did not undergo any significant changes relative to pre-treatment, with some of these animals actually increasing in oxygenation rather than the ones that decrease. Now, in order for us to actually understand what happens to these tumors, we have to look at them histologically as the study is terminated. So what I'm showing here is a representative CD31 and FITC stain section of our tumor that has been treated with Haddox and sacrificed at 2 hours post-treatment. If I zoom in and show you a cross-section of a vessel here, with CD31 being the staining shown in yellow, and the FITC-lectin shown in green around, what you'll notice is that surrounding most of these vessels, you will have a significant amount of FITC-lectin that has leaked outside of the vessel. So FITC-lectin is a marker of vascular perfusion. And if we look at the overall tumor, and of course, on average for all of our tumors, we see a significantly larger degree of FITC-lectin, or a FITC-positive tumor, on our treated animals in relation to our untreated control. And this is, of course, significant at the 2-hour time point, where we observe on average 40% more vascular leakage. And we suspect that because these vessels are no longer intact, the red blood cells, which are the sources of our photoacoustic signal, are leaking outside of the vessels. And because they're unable to now circulate back and become reoxygenated, they're simply staying in the tumor interstitium in a hypoxic environment, which is the reason why our oxygen saturation values dropped as much as they did at the early time points. And with that, I want to now talk a little bit more about the other approaches that we can use to look at the vessels of these tumors, and namely the fact that we can now also, in addition to monitoring the treatment response, we can also monitor the vascular growth of these tumors, which it could be thought of as the inverse problem, or the reverse problem, where now we have a tumor which is undergoing growth and not responding to treatments. So it is well known that as tumors undergo angiogenesis, their vessels become very chaotic, and that differs significantly from the normal counterparts. And our Cepstral analysis approach, which once again relies on the frequency content of our signals, allows us to monitor this vascular growth. So to do that, we initially developed some vascular tree models in order to study these tumors as a function of size. So the size of the tumor, we modeled it as increasing in size of the vessels, and these are the sort of representative three-dimensional views of these vascular trees and the corresponding photoacoustic images. And I remind you again of our 3 approaches, right? In the temporal domain, these photoacoustic signals look more or less the same, depending on which aspect or which cross-section of the tree you're looking at. In the frequency domain, once again, there aren't a significant degree of changes because the size of the vessels is the same. But in the Cepstral domain, we actually start to see differences between these tumors. And those differences arise on the location of this most significant Cepstral peak, which allows us to now identify whether or not there are periodicities in the structural organization of these seemingly random vascular trees within tumors. So we performed some preliminary experiments. This is still ongoing work where we inoculated these tumors and we watched them grow and imaged their vasculature at 27 and 43 days post-inoculation. And if we look at the Cepstral results for each of these animals, what we observe is a drop in that Cepstral peak, in the location of that Cepstral peak, as a function of time. And as these tumors grew larger, the location of that Cepstral peak grew smaller, which we suspect at this point, and this is still ongoing work, as I mentioned, is related to the spatial organization within the seemingly random vascular trees within the tumor. And for the few remaining slides of my talk, being cognizant of time as well, and I want to give you guys an opportunity to ask some questions at the end, I'll be talking a little bit on another type of therapy which photoacoustic imaging can monitor, and that is the combination of radiation and nano/microbubble therapies. And the idea arises from the fact that microbubbles, as well as nanobubbles more recently, are a source of acoustic contrast due to their large degree of acoustic impedance mismatch due to the presence of a gas inside a lipid shell. And ultrasound contrast agents, their behavior has been studied significantly when they are in the presence of an acoustic field. And from a clinical perspective, they also are extremely useful in allowing physicians to visualize abnormalities such as this carotid plaque here, as well as the vascularization of these plaques. So from an imaging perspective, they have a significant role to play in ultrasound imaging, but the application that I'll be focusing on is a therapeutic one, where instead of imaging these nanobubbles and microbubbles, you're actually bursting them. And that burst, once these bubbles are inside the vasculature, once again, the burst is as a result of the acoustic field that accompanies their injection. That burst actually causes vascular damage. And the vascular damage, it is hypothesized, causes the release of a lot of reactive oxygen species, which are in a way ideal for the radiation therapy that is to follow these treatments. And if we compare the degree of cell death that occurs as a result of a radiation alone or radiation and bubble treatment, we observe significantly more cell death in a radiation and microbubble treatment than we do in a radiation alone. So with that in mind, we designed a study where we're actually examining how the SO2 changes as a function of various treatment groups here, denoted by the x-axis on this curve. So what we actually observed is significant drops in the oxygenation of these tumors as early as 2 hours, but be further amplified at 24 hours post-treatment when we combine nanobubbles, ultrasound treatment, as well as a single dose of 8 grays of radiation. And that change is also different, or very different, from the change that we observe in the tumor oxygenation as a result of a single treatment of radiation, which actually has the complete opposite effect, most likely as a result of the inflammation of these tumors. So if we now look at how effective this marker is in predicting the degree of cell death, if we plot the percentage of tunnel, which is a marker of apoptotic cell death, as a function of the change in the SO2, what we observe for our single dose of 8 grays of radiation, we actually observe some cell death, but this is accompanied by a positive increase in the tumor oxygenation as a function of treatment. As we now combine these treatments, what we observe is significantly higher degree of cell death for those same tumors that had the largest drop in oxygenation, much like what is shown on the panel on the left-hand side here. So with that in mind, this is some very preliminary results that we will be following up on. With that in mind, I want to end my talk by linking everything together for you one more time. And hopefully I've managed to convince you that these radiofrequency-based photoacoustic biomarkers of treatment response, whether they are used for the studying vessel structure, vessel density through the envelope parameters, or the vessel oxygenation have the ability to be measured over time sequentially in a non-invasive fashion. And I hope that our results convince you that they can be linked to the success or failure of the therapeutic effect, regardless of what that effect is, whether it is a radiation and microbubble treatment or a thermosensitive liposome treatment, as I showed with the hat docs. So with that, I hope that you now understand, or perhaps are somewhat more convinced, that there is a link between thunder, lightning, orchestras, and tumors. And that link is photoacoustic imaging. And I would like to take this opportunity to acknowledge the funding support of various funding agencies, and of course the help of various individuals across many institutions here in Canada, as well as the United States with the nanobubble work. And of course, once again, thank VisualSonics for the opportunity to share some of my work, and I would be more than happy to take any questions from you guys. So Drew, I'll pass that on to you.
Some of our customers may be used to, we provide a lot of analysis tools on the system for various different application areas, but this is kind of getting a little deeper into, the data that underlies some of the images that come up on the screen. And I think I'll just sort of jump in with a couple of questions. First, one sort of obvious one is, how you actually do this on the Vivo systems, how you go about collecting that RF data.
So that's a very good question. I should have perhaps clarified. It says, as you mentioned earlier, the Vivo laser systems have both the Vivo laser as well as the new release, the Vivo Laser X, have something called the RF mode, which is my favorite mode, as you might guess. And this RF mode allows you to have access to the pre-beamformed RF data for both the ultrasound as well as the photoacoustic modes. And the exporting of this data away from the system is precisely why we've been able to develop a lot of these approaches that I was discussing. So it's safe to say that without access to that architecture on the beamformed and pre-beamformed RF data, none of these analysis techniques would be possible. And the Vivo Laser RF mode is the mode that allows you to do that quite easily, in my opinion.
Great, thanks. Another question that's kind of related to that, the techniques that you're doing, so that's how to get data off the system. The techniques that you're actually doing, could they be performed, by anybody, or how would that work if someone wanted to translate some of these into their own work?
Right. So anybody can access the data. That's the most important step. What's important to realize is that a lot of these approaches are derived from the well-established ultrasound tissue characterization community, and have been used and published and validated for a number of years. That the novelty here is now the application of these very same techniques to the photoacoustic imaging community, which typically relies on just the amplitude information that the photoacoustic data provides, while ignoring the frequency-based approaches. So can they be used by anybody? The theoretical answer is yes, but some knowledge of the underlying physics behind the acquisition of these signals, and most importantly, the theoretical predictions on what these parameters are supposed to represent, whether or not some of them are sensitive to size, or some of them being sensitive to concentration, or some of them being sensitive to both, are a good way of knowing whether or not they do work for your application. So we recently, a few months back, published a paper on some of these techniques in photoacoustics, and I referenced it. I'll go back to some of the slide where I had the reference. And I would encourage you to have a closer look into that paper, particularly focusing on how the frequency techniques that I was referring to, how they actually they can be implemented. So this is the reference right here in the bottom of slide number 9. And if I'm not mistaken, these slides will be shared with you later. So I encourage you to look at that in some more details to dive into the specifics of this, which unfortunately I don't have a lot of time to go over with, but they're pretty well explained in our paper. I hope that answers the question.
Yeah, that's great. Thanks. I appreciate the reference as well. You mentioned validation. How are some of these, tissue characterization techniques validated? Is it against histology, or are there other methods to do it?
No, that's a very good question. And the answer is depends on the application and what histology can or cannot tell you. So the most simple approach, so from a very pure physics standpoint, in order for you to understand, for example, how the envelope statistics parameters would change as a function of absorber size, the most easy validation would be one that involves tissue mimicking phantoms, which are phantoms that you can make yourselves. And there's a lot of recipes out there and quite a lot of work being done in the community these days to actually try to come up with a standardized way of coming up with the perfect photoacoustic imaging phantom, reference phantom that is, that allows you to now study these parameters in isolation in a well-controlled environment in order for you to understand how sensitive your system is at differentiating, say, various sizes or concentrations. Now, histologically speaking, when it comes to biological or biomedical applications such as cancer treatment monitoring or other types of applications, histology still remains the gold standard. And histological slices can be co-registered with your photoacoustic acquisition, provided you know where you performed your imaging relative to the retrieved histology. And if you wanted to study, for example, sizes of vessels or distribution of vessel sizes, simple to use stains such as CD31 can be used to provide information with regards to how big or how small their vessels are, or how does the size distribution of those vessels changes as a function of treatment, and then bring these parameters back to your quantitative biomarkers and then perform more direct calibrations or rather validations of what it is that your imaging is telling you.
Okay, great. Yeah, and I was sort of, where histology fits in with, I guess you're correlating your histological slices with the data you end up with, and then that you've sort of tried to validate with the phantom. You did mention the phantom. I want to ask how well that phantom matches the in vivo situation. In other words, how well do beads simulate blood vessels of different sizes? It's really the blood that we're imaging with photoacoustic imaging. Just wondering if you can comment on that.
That's a very good question. So it all depends on also what the application is. So in other words, if you're to ask somebody, is a vessel the same as a bead? The answer is no. But from a very, very theoretical approach, if you imagine within your imaging resolution volume, you have a very, very large number of absorbers, and each of these absorbers has a different size. What really the mimicking or the resemblance of your phantom to your biological system will depend on what exactly are you trying to mimic. If you're trying to mimic, say, the concentration, then it would be a very simple way of checking whether or not the concentration of your vessels is the same as your concentration of bead sizes. Concentration of beads of various sizes. So looking at the number of vessels that you might have per resolution volume might actually be a very simple thing that you can do with one of these phantoms full of beads. Now, if you want to study things like how perfused your vessels are or what their oxygenation levels are, then perhaps static glass beads that have been dyed with a black ink are perhaps not the way to go. And at that point, you might want to consider other types of approaches where you are using phantoms with blood, with whole blood or oxy or deoxy blood. So where you're now looking at vessels of sizes that you can control and make them as small or as large as you need to mimic the say vasculature of whatever it is that you will be studying. So to answer your question, that the bead phantoms are good in order to retrieve the frequency response of your system, making the assumption, which is not a terribly bad assumption, that you have a large number of absorbers within your resolution volume, and at the same time, the sizes of these beads reflect whatever it is that you are studying, such as tumors.
Thanks, Eno. That's a very thorough answer to that question. I know there's a lot of work going on, as you mentioned, in the photoacoustic community to make a phantom that can be standardized so it can be sort of used across different systems to be able to measure different parameters. So yeah, that's great. We've got a question here coming in asking about bursting of your micro/nanobubbles. They're saying, is it caused by the frequency that you choose for the photoacoustic imaging? And I would say that they likely mean the ultrasound, that it's the ultrasound that you're using to burst
Correct. So it's a very good question, actually, and one that people ask all the time, because when we talk about the concept of frequency, someone might attribute that to the optical wavelength frequency, which is the frequency of our light pulses. But you're correct in saying it is precisely the frequency of the incoming sound wave, and not just the frequency, but also their pressure, because there's a lot of work that's been done in the ultrasound community over the years that demonstrates how both the frequency and pressure, and sometimes pressure-dependent frequencies, have the ability to make these bubbles not only oscillate but also burst. So in our specific example, because it's a therapeutic application, it is an external single element transducer operating at 570 kilopascal and 500 kilohertz, which is a frequency that has been determined for the sizes of these bubbles, both the nano and the micro bubble, to be the most optimal frequency that actually causes these bubbles to burst. So we are not imaging these bubbles with the Vivo laser system and transducer at all. It's an external system that actually burst these bubbles before we apply the radiation, at which point we bring them into the Vivo laser system to image the impact of this treatment. So I want to make it perfectly clear, you can image them for a diagnostic standpoint with a Vivo laser systems, and you can look at some beautiful images of a vasculature through contrast mode, but in this case we're simply bursting them externally with a single element transducer for therapeutic purposes.
There's a follow-up question here asking about, is there a limiting frequency? Is there a frequency beyond which, or a frequency or a pressure even, beyond which you're damaging healthy tissue? And I'm sort of, I think the implication is, is there, a mode of ultrasound that you're using that will burst the bubbles but not damage healthy tissue?
Right, so the pressures that we are using here would fall under that regime. So the damages that we are inducing as a result of the bubbles oscillating, you have to also keep in mind that these bubbles are internalized in the vasculature of these tumors. And the vasculature of the tumors is, of course, surrounded by tumor parenchyma. So when we talk about damage to healthy tissues, we're only insonicating the tumor. The tumor is a subcutaneous tumor, so it's super easy to localize within our focus. So as far as the surrounding healthy tissues in the leg muscle or other tissues that this work has been done on, such as the mammary fat pads, they typically are spared from the effects of the ultrasound, and the ultrasound is only applied onto the tumor. Now, the ultrasound itself, without the presence of bubbles, there are frequencies and pressures, mostly pressures, which actually will cause irreversible damages to tissues. So, and I'm talking right now about approaches such as high-intensity focused ultrasound or histotripsy approaches that actually, with the pressures of the sound waves, will damage the tissue. That is not what we're doing here. So our pressures are selected in such a way that they're only meant to burst these bubbles and not cause irreversible damage to the surrounding tissues.
Great. Yeah, I think that answers the question quite nicely. I want to come back a bit to your RF-based techniques. One question, I guess, is, let's say you're using your RF techniques to measure something like vessel density. do you see some of these techniques being standardized into, standard measurements that are used in ultrasound in the future?
Well, that's a hope, really. That's one of the motivations behind this work. But what we also need to keep in mind is photoacoustics has the ability to image multiple biological length scales, right? So if we are now performing what is typically known as optical resolution photoacoustic imaging that generates those beautiful images of tumor vasculature or even surrounding healthy tissue vasculature, at that point you can actually visualize these vessels, and you can do that with very high spatial resolution, but you're limited with the penetration of light because you're now actually focusing the laser beam. So the approaches that I discussed here today are typically meant to address challenges in acoustic resolution photoacoustic imaging, which is a type of imaging where you are now illuminating a larger volume of tissue with a diffuse laser beam, such as the one that comes out of the vivo laser transducers, and at the same time you are relying on the frequency of your ultrasound to dictate the spatial resolution of your images. So as you saw from most of my images that I showed in this talk, we can actually resolve individual vessels, but not as well as you would in acoustic resolution photoacoustic imaging, because the resolution of our system is band limited. So our approaches, and the one you mentioned with regards to monitoring vessel density, they're precisely tailored towards these applications where you're unable to generate these beautiful images of the vessels, but you still, using those speckle filled images that I showed, are capable of analyzing the frequency information and, most importantly, the physical parameters of that frequency information encodes, one of which being the vessel density or size.
Great, thanks so much for that. We're just about out of time. I've got a few things to kind of wrap up. I do want to thank you, Eno, so much for this work. It's really interesting work. It really is sort of digging beyond the surface of the images that are there and getting into the data that really makes up those images, and finding these new techniques to be able to essentially do new measurements. One thing I want to point out is the SO2 histogram, some of the work that you showed there. And for everyone who's on the line, Eno was actually the one who suggested, hey, you guys should really have this measurement in your software, because, Eno, you've been doing it, sort of offline. So we actually added that into our new software release, which we just had about a month ago. So we now have the ability to draw a region of interest around your SO2 or oxyhemo image and generate that histogram to be able to measure things, like you mentioned, like heterogeneity of tumors, looking at the mode, looking at different aspects of the distribution of SO2 within, let's say, a tumor. So that's in there. You'll be pleased to hear. I also want to mention that for anybody who is using a Vivo system and who is collecting RF data, or who wants to collect RF data to maybe try out some of these techniques, we do have a guide which can guide you through. We have some scripts which so you can read this data into MATLAB. So please do reach out to us. You can reach out on our website, www.visualsonics.com. You can submit a question or request via our web form. So that would be a really great way of asking us about this RF data documentation. Please check out our other webinars. This one will be posted online. We've got, I think, over 30 webinars now online. You can go back and look through the catalog and look and listen. And of course, you can find us on our social media channels. And to contact us, you can go to visualsonics.com/support/customer support to get in touch with us. Again, Eno, thank you so much for your time today and for presenting your great work. And until next time.
It was a pleasure. Thank you so much for having me.